Okay, this lecture is going to be on hemostasis. And hemostasis is the clotting system that occurs within the body. Also, we'll refer to it as a clotting cascade, we'll talk about, which is a part of this whole process of hemostasis. But the first thing we have to do is understand the difference in the blood type makeup that has these clotting factors in it. For example, if you were to take a blood sample, like I would have here in sample number 1, you take the blood, just pull blood from someone, put it in a test tube without an anticoagulant, and it clots. You're going to have the blood clot at the bottom of that, which has the clotting factors in it. Above that would be the buffy coat, and then that would be serum, the clear fluid that would be above it. If we take, as in test tube 2, which has an anticoagulant in it, and that anticoagulant then, this would be an anticoagulant that's in that. And what it would do would be it would keep the blood from clotting, spin it down. This would contain red blood cells, and then this would be plasma. So serum and plasma, even though we use them interchangeably sometimes, are not the same thing. Plasma would have your clotting factors in it, serum would not. We also need to understand that in the clotting mechanism that occurs, there are 2 types of mechanisms. First we have is the intrinsic mechanism, and then the extrinsic. Now, what's the difference? On an intrinsic clotting mechanism that occurs, that's clots that form within the bloodstream without having any external trauma or anything inserted on the vessel. So that would be things like your deep vascular thrombosis, clots that occur within the heart that create myocardial infarctions, plaque that builds up and creates the clots that occur where we have these aneurysms that occur in the bifurcations at the carotid artery, the internal iliacs, or even around the heart. So those are intrinsic, they're internal type clots that occur. And they have a complex clotting mechanism that goes through these. So you would have, in the intrinsic pathway, you would have clotting factor number 12, which is activated, and then it converts inactive 11 to active 11, which goes to active 9, which goes to active 8, which goes to 10, active 10. And then we'll have what is called a common pathway. This would be the common pathway, which is in both of the intrinsic and extrinsic pathways. The extrinsic pathway would occur when we have things like an insult of a knife wound, a stab wound, cut your finger, that type thing. It's extrinsic, external, and it traumatizes the lining to the vessel, and that stimulates something that is known as a tissue factor, which is number 3, clotting factor 3, which becomes active, which stimulates inactive 7 to become active, to activate inactive 10 becomes active 10. And then active 10, whether it be intrinsic or extrinsic, then through factor 5, which stimulate factor 2. Factor 2 is inactive form is prothrombin. It converts it to the active form thrombin. Thrombin then converts active form of fibrinogen, which is inactive fibrin, to the active form of fibrin. We're going to walk through all of this individually to show you what those are. So when we look at these, then my factor 2 is going to be inactive, would be prothrombin, and then activated it would be thrombin, which would convert my inactive form of factor 1, which is fibrinogen, and it would convert it to the active form of fibrin. Okay, now just keep those in mind, and here's the way you can remember these. All right, and then when we walk through each of these steps of it, you'll have a better appreciation for what takes place. But the way to remember these is in my intrinsic, it goes 12, 11, skip 10, 9, 8, and then to 10. Okay, and 10 is the central point. Okay, so it goes 12, 11, just count backwards, skipping 10. 12, 11, 9, 8, and then 10. Okay? How do I remember the extrinsic? Well, it's tissue. The tissue factor is 3. 3 plus 7 equals 10. So 3 plus 7 equals 10. Okay? Now then, how do I remember the common pathway? Well, 5 times 2 times 1 equals 10. Okay? That's just a, take it or leave it kind of thing, but it's a short way of remembering these factors. And we're going to look at, okay, how do those factors come into play now in our clotting cascade, and how do they do this? Now, another thing you also need to remember about these clotting factors is that some of them are vitamin K dependent, and that becomes very important because it's produced, vitamin K produced by the liver. If we have liver diseases, then we don't have the vitamin K, then it impacts our abilities to have clotting, so that becomes significant. And factors 2, 7, and 9 are vitamin K dependent. And without vitamin K, whether it be intrinsic or extrinsic, it's going to inhibit the abilities to form a clot. Another thing that's important is calcium. If we have calcium deficiencies, it also is going to interfere with clotting factors. And calcium is required in every step except the first 2 steps of the intrinsic pathway. Other than that, all other steps require calcium as an activator to stimulate activation, in other words, of the inactive form of the clotting factor to the active form of it. So calcium becomes very, very important, and so does vitamin K. All right, now then, let's get a layout of what we're going to look at on the blood vessel itself. And so if we look over here at the blood vessel that I've drawn here, and I apologize for the drawing, but I think at least you can get the concept that I'm after here to get this across. Because we've talked many times about the fact that you have an intima, a media, and an adventitia. The intima is the inside lining of this. The media is going to be this portion of it that's going to contain from here here. It's going to contain my smooth muscles, and it's going to contain the area of collagen, which is right below the endothelium, and it also contains these nerve cells, which are nociceptors. So I would have intima, media, and adventitia. Adventitia is the thick fibrinous lining around the outside of any vessel. So the inside cells then, this is going to be endothelial cells. These cells here are endothelial cells. Immediately beneath that are going to be these collagen fibers that I've got in blue here. Those are collagen fibers that are there. And then immediately below that is going to be these layers of smooth muscle that you're going to see. So it have smooth muscle. And on the surface of the smooth muscle, there's receptors on these we'll talk about in a moment, because that's how they get stimulated to contract. There's a receptor on each of those, but there's also a nerve cell on those that are called nociceptors. These nociceptors are pain receptors now on the surface of this smooth muscle. So if we look at that, that's going to be, if I'm looking at it cut into and I'm looking at the end of it. And so now we're going to draw this out longitudinally, and then we're going to look at it from a side view and then be able to walk through each phase of the clotting mechanisms, okay? Okay, now when we look at the clotting mechanisms, and I've written up the top, there are 5 steps. And a lot of times you'll just see this written as 4 steps, and they don't include the step number 5, fibrinolysis, as a part of it. But it actually is a whole part of the clotting cascade in the fact that it's the remodeling, reabsorbing of the clot. But as far as the clotting cascade or the clotting mechanisms itself, it includes the first 4 to get the clot formed. And that's number 1 is going to be a vascular constriction or vascular spasms. You'll see it written both ways. And by the way, you will see these 5 things written in the book on Guyton's. He classifies these a little differently as far as terminology, but the most general accepted consensus for the clotting mechanisms is, as I have it written here, it's vascular constriction, then the formation of the platelet plug, blood coagulation. And then number 4 is clot retraction and repair. You'll see that written as remodeling or restructuring of the clot and so forth. And then step number 5, which we'll include in this as well, is fibrinolysis. And the reason for doing that is that's the way the clot's going to be reabsorbed. And we need to cover that because it does have some significance as far as therapeutics are concerned. Okay, so let's look at these, and we're going to take these one step at a time. And the first thing we're going to look at is vascular constriction. Okay, so what does that mean? Well, when we talk about vascular constriction, if we get some type of a trauma to the inside lining of the wall, if you'll remember when in the gross anatomy, and we looked at the fact that we had the common carotid artery that came up and split into the internal external carotid artery, and we talked about having that little nidus in the center, that little division of it. When the blood hit that, it would create turbulence, and if high pressure, increased velocity, so a person has hypertension, that enhanced that turbulence, and it would over time create trauma to the inside lining of the wall. Well, that's what we're talking about, those kinds of things that could happen. So we create some trauma to the wall. So what we've done is we've created some trauma to this endothelium. So if we come here and we look at it, so let's go in here and let's create some trauma to this endothelium. Let's just erase this center portion right here, and we've created trauma. And here we've opened this up. And so if we do that, now what we're going to see is that by doing so, I've traumatized that endothelium. And you remember from the studies that we did on things that would cause vasoconstriction and vasodilatation, there was a thing that was called endothelin that was produced by the endothelin, and it causes vasoconstriction. And that's what's going to happen here. Okay, so we're going to have roughly 3 things that are going to impact this to cause this vascular constriction. And the first one is going to be production of endothelin. And endothelin, when it is produced, you're going to see that it's going to be produced by the cell here. And this endothelin, when it is produced because of this traumatized cell here, what's going to happen is it's going to stimulate these receptors. You remember I told you that on these smooth muscles, we've got these receptors that are on here. And it's on all of them. So this endothelin then hits that smooth muscle, and it's going to cause vasoconstriction. Okay? Now, another thing that happens when it does that is there is known as a myogenic response. If you remember back in some of our other lectures, we've talked about that type thing before, where we'll have a myogenic activity that will occur in some of vasoconstrictions. And that's what happens here is when that stimulates, you will get this myogenic response. And a myogenic response, again, is simply because it's been stimulated by the activity of the endothelin that's on those receptors that causes constriction. And once that happens, it initiates a response within the other smooth muscles to cause constriction to occur. And then the other thing that you're going to have happen here is you're going to have a neurogenic response. And that's going to be because, again, when you have this endothelium, the endothelium in there that's disrupted, then it's going to go and hit these receptor sites on these nociceptor cells. And those nociceptor cells, again, are pain cells. And so what you get then is these nociceptors response. And that nociceptor response is because of the stimulation on those endothelial cells. And all of those are going to cause vascular constriction and get this whole area. And it's trying to pull those endothelial cells back together to fill this defect and to stop the bleeding. Think about it. This whole process is to minimize, decrease, or stop bleeding. And so the first thing that's going to happen is when you get trauma to the endothelium, it's going to try to close it off. It's going to try to to stop it so that it can stop the bleeding process. Now, the second step we want to look at is going to be step number 2. Let me put this in blank here. Step number 2 is going to be formation of the platelet plug. And how does that happen? Well, okay, when we look at this again, we go back down to our traumatized cell where it's injured. Endothelin, there's a protein that we have that is in traumatized endothelium on the bottom side of it. There is a protein that's normally there, and it comes into play with traumatized endothelium, and that is known as von Willebrand factor. Okay, and so these two things will pop up here, and those are going to be the von Willebrand things. And what they'll do is they will have a receptor site on them. So they've got this receptor site that is located on these cells that are here, and this is a glycoprotein type receptor site that's there. And by the way, those glycoprotein receptor sites are important because that's what binds the platelets to them, but it also comes into play in some of the drugs that you use, especially von Willebrand's, or where you have a situation where you use a drug that will stop the clotting from occurring that interferes with that. And remember, we've got throughout this area, we've got these small little fragments of platelet cells that are all over the place. They're all in the blood. They're going through here constantly. You've got these little platelet cells that are there. And so what happens is when the von Willebrand factor is there, that's what this is the von Willebrand factor. Von Willebrand factor. It's what these are. So the von Willebrand factor that's there has the receptor on it, and once it is initiated and activated, it draws these platelets to it. So these platelets are going to be drawn to the site of trauma that's there, and so what you'll have then is you'll have all these platelets that'll come in, and they're going to be drawn to it. Now, it also stimulates, because of these platelets, it stimulates production and release of 3 chemicals. And the chemicals that you're going to have released for that is going to be ADP, adenosine diphosphate. And then you're going to have also, you would have thromboxane A2, and then you would have serotonin. Okay, these are the things that are going to be released. These 3 chemicals are going to be released, and these are important because you also comes into play in some of the drugs that work on them. But these platelets, once they are drawn to the von Willebrand factor and attached to the receptor site, stimulate the release of these chemicals. Now, ADP, thromboxane A2, what they do is they work together so that they go up and they stimulate, both of them together in combination will go up and also stimulate platelets to be drawn to the site. So you just increase more and more platelets combining here. And this once that stimulates that to get these platelets to form on here, what that's known as is platelet aggregation. Okay, so that's going to be platelet aggregation. And that is when ADP and the thromboxane A2 stimulate those platelets, and they are released because of the initial platelets that were bound to that von Willebrand factor. And once that happened and stimulated other platelets come to it, then they produced the chemical ADP and thromboxane A2 and serotonin. They then, in turn, the ADP and thromboxane A2 go and stimulate the platelets to stimulate them to be drawn to the site. So that is known as platelet aggregation. It's pulling them together and aggregate them in one area. Another thing that happens through this is that thromboxane A2 and serotonin act just like, you remember we had the receptor site where we had the endothelin that goes and attaches to the receptor site on the smooth muscle here and stimulated vasoconstriction. Well, thromboxane and serotonin do the same thing. So they enhance that vascular constriction or vasoconstriction of that pulls that together, that tries to stop the bleeding by enhancing that. So thromboxane A2 and serotonin work in conjunction to go and stimulate those receptor sites on smooth muscles to increase or enhance vasoconstriction. The adenosine diphosphate, ADP, along with the thromboxane A2 go to stimulate the platelets to be drawn to the injured site of the endothelium, known as platelet aggregation. Okay, so all of that process that we just talked about there then is the formation of the platelet plug that we got together. Also, I want to reemphasize to you, those areas become important because of the von Willebrand and the receptor site there, that glycoprotein that's there. Also, glycoproteins will bond together. There's glycoproteins that come into play in not only binding those platelets to the von Willebrand factor, but also in binding platelets together, cross-linking them together. And why that's important is there's drugs that will come into play in preventing that glycoprotein bridge from forming there, and so they can't clot. Okay, so now we're going to look at step number 3. So we've gone through step number 1, in which we had the vasospasms or vasoconstriction. Step number 2 is where we had our platelet plug formed. And so now we're going to look at blood coagulation. So if you remember back from our platelet plug that was formed, we had, once the endothelium was damaged, then we had these von Willebrand proteins that were stimulated, and these von Willebrand proteins had these receptors on them. These are glycoprotein receptors, and they would attract platelets that would come, and the platelets then would be drawn and formed and attached to those receptors. They would in turn attach and draw other platelets, and they would stimulate the chemicals of ADP and thromboxane A2 along with serotonin. And we mentioned about what all those things did. And they form this platelet plug that's going to be drawn out here. And this would be our plug that's formed. All these platelets are going to build upon this thing, forming this plug that's there, okay? Now, if you will remember the clotting factors I drew out for you at first, you had an intrinsic and an extrinsic clotting factor system. We're going to look at the intrinsic to begin with, and then we can bring the extrinsic into play so that you can understand that. So if you will remember the numbers I did, and by the way, most of the time I wrote that in regular numbers, but you'll see it written in Roman numerals most of the time. But what happens once you get this platelet plug formed, it creates, due to reaction with other chemicals, it'll form a negative charge around this plug, okay? Now, you have to remember, all of these plating factors that we talked about are made by the liver, and they're flowing throughout the bloodstream constantly in an inactive form. So you've got factor 12, 11, 10, 9, 8, 7, 6, 5, 3, 2. All of them are normally in the system. Fibrinogen, prothrombin, all of those things are naturally just floating around within your blood, but they're in an inactive form. For them to be functional, then they have to be converted to an active form. And so what you will have is that when this plug is formed, this platelet plug that's formed here with all these platelet cells around it, it creates this negative charge around the surface of it. And what happens is the first protein that comes in contact with that is going to be factor 12. And factor 12 comes along and it's an inactive form here. And then once it hits into this negative charge, then 12 becomes active. So 12 is now active, okay? So what 12 does, and by the way, again, you write this in Roman numerals as well as what you'll normally see it written in, and I'm going to keep it in this format simply so it relates back to what I drew out for you earlier. Inactive 12 then hits these negatively charged that has been set up by this platelet plug, and becomes active 12. Active 12 then will go and react to the 11 clotting factor, which is inactive, and it activates it. And so now 11 is activated. 11 then will react to inactive 9, so I would have 9 is in active form. It reacts to it and becomes active 9. Active 9 then will combine, you'll have 8, which will go and combine with this one, and it activates that to activate 10. Okay? So 10 now was inactive. You had a 10 inactive, and 8 in with 9 creates 10 to an active form. Okay? And remember, we talked about earlier also, calcium and vitamin K are important in a lot of these, like within 2, 7, and 9 is vitamin K. And then also in all these processes, calcium is important in these. Once 10 becomes activated, then 10, along with factor 5, remember 5, as we drew out on earlier, is now I'm in a common pathway. If I'm in the common pathway, it's 10, 5, 2, 1. Remember that from the formula I did for you earlier. 10, 5, 2, 1. That's clotting factors. Well, 10 reacts with 5 to stimulate 2. Okay? Now, you have to understand that 2, in its inactive form is prothrombin. And then with 10 and 5, it converts it, the number 2 here, let me write it as written before so that we keep in consistency here. Number 2 then is in its inactive form is prothrombin. Prothrombin is virtually inactive too. And it's going to convert it to the active form of thrombin. And thrombin is active too. Okay? Thrombin then will go to convert fibrinogen, which is inactive 1, so it'll go to, this is 1, and its inactive form is fibrinogen is inactive 1. and it's going to convert it to the active form of fibrin. And that's active 1. Okay? All right, so what you'll see happening then is this whole area from 10 is the common pathway. That's going to be in either intrinsic or extrinsic. The intrinsic is going to go just like that Y that I drew out for you. Had the 10, 11, 9, 8, 12, 11, 9, 8, 10. Then it went 5, 2, 1. And the extrinsic had 3, 7, 10, 5, 2, 1. Go back to that first slide and look at those, and you'll see those drawn out. And remember that, because now we're taking the extrinsic side of this. Once the platelet plug forms, it creates a negative charge around it. These clotting factors are produced by the liver, and they're floating around in the bloodstream. 12 then, in the inactive form, comes and it hits these positive charges, and it becomes active 12. 12 then initiates inactive 11 to active 11. 11 will take inactive 9 and convert it to active 9. 8, and combined with active 9, will convert inactive 10 to active 10. Active factor 10 combines with 5 to take factor 2, which is the inactive form, is prothrombin, and converts it to its active form of thrombin. Thrombin then can go and convert the inactive form of 1, fibrinogen, to fibrin. Okay, so now we have fibrin formed. We have one other step that comes along here. There's one other thing, and you have to realize that fibrinogen, when you look at it, fibrinogen is just free floating out here like this. It's just out here on its own. You got fibrinogen just free floating out, just like you have factor 12, factor 10, it's just free floating individual. But when it becomes active to fibrin, what that does is it pulls those together, and it makes these solid chains of these things. Okay? It'll make these solid chains like so. And these solid long chains. Okay, well then what happens to that is there is a factor 13 that comes along and comes in contact with the fibrin, and what that does is it forms a mesh. What does that mean? It forms a fibrin mesh. And a fibrin mesh is simply, it means it takes these long chains and it connects them together like so. This is real similar to the same things I do with polymers in the plastination lab in making them into plastic. Is I'll take these long chains and then put them through a chemical process that links them together like this, which does the same thing as factor 13. It forms them together in a mesh, and then it makes that polymer then plastic. So that's what happens. Factor 13 then comes into play to take, this would be normal fibrinogen, and it forms fibrin, which would be one long chain, and then when it comes in contact with factor 13, then that forms it into this network of mesh known as a fibrin mesh. And so what would happen then is if you look at this and draw this out, it forms a net over this clot. This fibrin meshwork forms this net over this. so that it'll hold it in place and it can't just free float loose into the bloodstream, so it will hold it down there. Okay, so that is your clotting cascade for your intrinsic clotting system. Okay, so if this is the intrinsic type, well then how does the extrinsic come into play? Okay, so let's look at that. Okay, so let's say if I look at the top portion of this, what I've done is I've drawn it like I've got a knife wound here that has occurred. And so that if I have this knife wound that has occurred within this, that's gone in and stabbed this area, punctured through this portion of the endothelium, when you get trauma of that, you stimulate the tissue releasing factor, which is number 3. So what you're going to get is you're going to get, with that trauma created to the endothelium, you're going to get all the other things to release just like we talked about, but you're going to get tissue factor 3 released, also known as 3, okay, that's there. And tissue factor 3, go back to your chart again. Remember what tissue 3 is going to do? It's going to go react with my 7, which is inactive, and it's going to convert it to active 7, which can really do 2 things. It can go directly to 10, and what it'll do is it bridges right in this area. It goes and it converts inactive 10 to active 10, and then the whole network follows out. Or it can go over, this can go over and stimulate this portion between 9 and 10, but the results are the same. In essence, what's going to happen when you get trauma to the endothelium, it's going to stimulate tissue factor 3 release. The clotting factor 3 is known as tissue factor, and it's released due to traumatized endothelium. And when it does that, it converts inactive 7 to active 7, which active 7 then goes directly to inactive 10 and converts it to active 10. Active 10 with 5 converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin. Fibrin then will come in contact with factor 13, causes the fibrin mesh or net to occur that goes over the clot and holds it in place. So that is your whole theory of your clotting cascade that occurs within that. And if you'll go back again to that I drew out, and I'll draw it once again so that you're sure that you remember this, is the fact that if I'm going to look at my intrinsic, I'm going to start with 12. 12 is going to convert 11. 11 to 9, skip 10. 9, 8, and it's going to go to create 10. Then when inactive 10 converts to active 10, is going to go with 5. It's going to convert to 2, which is prothrombin to thrombin, which converts 1. Fibrinogen to fibrin. My extrinsic is the 3, that's the tissue factor 3. It's going to go to 7. 7 goes to 10, and that's going to convert to 5, which is going to convert prothrombin to thrombin, which is going to convert fibrinogen to fibrin. Okay? And the way to remember that is you just go in reverse order. 12, 11, skip 10, 9, 8, 10. Okay? Extrinsic is 3 plus 7 equals 10. How I know my common pathway is 10. 5 times 2 times 1 equals 10. Okay, and that will keep you on track as far as what clotting factors involved in intrinsic versus extrinsic. The extrinsic pathway is much faster. It's like within 10 to 30 seconds. I mean, it's fast, okay? the extrinsic is. Whereas the intrinsic, you're talking about anywhere from 3 to 6 minutes to be functional. Okay, so step number 4 is going to be clot remodeling, which is also referred to as retraction and repair. But in the clot remodeling, there are 3 processes or 3 things that take place, and we need to understand these because there's some chemical involvement that's associated with these as well that we need to be aware of. The first thing that happens in this clot remodeling repair is that the platelets, when you look at these platelets that are sitting here together, all in those, they will tend to contract or pull in. It's just like that they're grabbing each other and squeezing them together and pulling them together. So you end up getting this thing that is known as platelet contraction. And that's just the platelets themselves pulling together. So it pulls the ends of the endothelium together. It pulls the whole clot together. It's just like this fibrin mesh that's laying over the top of it is just contracting and pulling them in together. And it's a platelet contraction. The second thing that happens is that this platelet contraction, when it occurs, it stimulates a chemical to be released. And this chemical that's released is known as platelet derived growth factor. Or also known as platelet derived growth factor. Okay? Okay, and platelet derived growth factor, what it's going to do is platelet-derived growth factor is going to go down, and what it's going to do is it's going to help in any of smooth muscle damage. In other words, if you had trauma to this area to the point that it damaged any of the smooth muscle, it will help enhance regrowth of that, or any of this collagen that occurs in there. It's going to help regrowth of that. It's just to reestablish growth within those areas. It'll enhance things that will occur like mitosis. It'll enhance endothelial replication to regrow and form fibrin and scar tissue and so forth within there. So that's what this platelet-derived growth factor is going to do. It also stimulates another chemical, and this one is known as vascular endothelial growth factor. Okay, and vascular endothelial growth factor, what it does is it just think of the word, what it says, vascular endothelium. It's going to enhance the regrowth of vasculature of the endothelium. It's going to regrow this endothelium that's here. Okay, and that's what it's going to do. So then when he does all that, then it's going to retract this thing. It's going to pull in the platelets, contracting, pulling them together. It's going to stimulate the release of 2 chemicals. One is going to be a platelet-derived growth factor that's going to stimulate regrowth of the musculature and the collagen and the things that are laying beneath in the media of the vessel. And then it's going to have a chemical release that is vascular endothelial growth factor, which is going to stimulate the regrowth of the endothelium that's there, so that you can remodel and regrow this whole area. And that is step number 4. Okay, now that brings us to step number 5. And again, this step is generally not included within the clotting and the clotting cascade because it is the way in which clots are dissolved. But it's a very important normal process that occurs in your body. So step number 5 in this is fibrinolysis. And fibrinolysis occurs, it's a way in which we dissolve these clots. In other words, if these clots form within our system, then to keep those clots from just to a point that they'll break off and cause problems in our body, we have to have some natural way in which clots are reabsorbed, remodeled, and degenerated within our body without causing any damage. And what you'll see is on the endothelial lining of this, you will have these proteins, okay, that are here. And these are normal proteins that occur on the endothelial surface, and these things are known as tissue plasminogen activators, or we run it as TPAs. I'm sure y'all are all familiar with that and heard of that. TPAs are tissue plasminogen activators. And how this works is normally in the system, you've got this plasminogen that is floating around within the system. And so this plasminogen that's in the system comes in contact with this protein, and it converts it to plasmin. And plasmin is it's kind of plasmin then is kind of like your Pac-Man. He's a gobbler. He loves to eat up fibrin, and he will come over and attack this fibrin that is on the clot. He loves fibrin, and so he'll eat up this fibrin. And what he'll do is he will release, when he dissolves this, he release fibrinogen, which, remember, it's normally within the circulation to go and it can come back to form fibrin again, and also releases a thing called D-dimer. And D-dimer is important because that is one of the tests that you'll run for people that have deep vascular thrombosis or you think there's a clot, because for there to be any D-dimer evident within the body, there has to have been plasmin available to break down a clot to be able to release the D-dimer and fibrinogen. So that's the reason that's important. That's the only way the D-dimer is going to be formed is if there's a clot that's been there and the plasmin has broken that down. All right, so now there's your 4 steps for clotting, and then the 1 step of fibrinolysis and resolution of the clot that comes into play. But now we need to look at, what keeps the blood from normally clotting within the system? Why doesn't the blood just clot all the time then? What keeps it from doing that? Okay, so let's look at that. What are the natural things that are within the system to keep it from clotting? All right, so now let's look at what things keep the blood from clotting naturally and keep us from having issues with blood clotting in us. Well, there are several things that do that, but basically 3 things. Number 1 is that the endothelial cells produce nitric oxide. Okay. And nitric oxide, if you remember when we've talked about it before, is a vasodilator, but it also enhances the ability of hemoglobin to be able to disperse and exchange oxygen better and so forth. But it also plays a role in being secreted by endothelial cells to play a role on the platelets. It also produces another thing, a prostacyclin known as PGI2. And again, it's a prostacyclin. And this prostacyclin, what it does along with nitric oxide, remember I told you you've got these platelet cells that are normally floating around the system here, okay? And those platelet cells, again, just like we've talked about in our clotting mechanism, are looking for this opportunity to go into to form a blood clot. But what it does, these two things here will go together, and they will individually or together deactivate those platelets. So they will deactivate them. so that those platelets then can't go and bind to an area. So it keeps it from forming the clots. Okay. The next thing we have is that number 2 is we have another type of a protein that is a receptor site that we see is heparin sulfate. Okay, so we've got a heparin sulfate here. And most of you are familiar with heparin and how it works. But what it does is it combines with an antithrombin 3 protein. So if we have this protein that comes along, it fits in here and it combines with it, and it's antithrombin 3 that combines with it. And then what that does is they will deactivate your clotting factors, and the factors that it would deactivate. So the heparin sulfate, then when it forms this protein of antithrombin 3, then what it will do is it will inactivate your clotting factors 2, 9, and 10. Okay? So what it does then is it inactivates those. So now they become inactive. So now those clotting factors are no longer able to play a role in causing clotting. Remember, 10 is one of the common pathways, so it's going to get extrinsic and intrinsic along with 2, which is thrombin. So heparin sulfate, nitric oxide are 2 of them. The third one that we'll see that plays a role in this, which is a little more complicated, is that you'll have another protein that is going to be on the surface, okay? And this protein is thrombomodulin. Okay, thrombomodulin is on the surface of the endothelium inside the vessels, and what it'll do is it will react then with thrombin. Remember, that is clotting factor 2. Clotting factor 2. All right. So when it reacts to it, then what it does is it will create a network that ties into, you have these proteins that are going around that are protein Cs. Okay, there's a protein C that are in circulation. And what it does when it comes in contact with that, then it makes this as an active protein C. So it'll come in contact with this and make it an active C, which inactivates clotting factors 5 and 8 and inactivates those. So it makes them inactive. Okay, so what you've done then is from step number 1, you've got nitric oxide and your prostaglandins, that PGI2s, that will inhibit the platelets or inactivates them or deactivates them so that they can't go and form a clot. Then you have heparin sulfate that combines with AT3 that causes 2, 9, and 10 clotting factors to be inactive. And then you'll have thrombomodulin receptor, which reacts with thrombin, which is one of the factor 2 that's normally floating around. And when it reacts, then protein C, which is also in the area, it will inactivate or activate protein C, which protein C in an active form deactivates or inactivates 5 and 8. So then you can't get clotting. So those mechanisms that you see there are what's going to keep the blood from clotting. Okay? All right, so that covers all of hemostasis. And remember, as we look through these first stages of it, as you get into pharmacology, there are going to be many drugs that are going to come into play that are going to have their action upon one of those sites in clotting. So get familiar with all those steps that we've carried through this so that you will be familiar with those when it gets to that phase of your pharmacology. Okay? God bless you and thanks.